extremely pleased to have the opportunity to introduce Professor Sarah Gehlert. But before I do, I want to echo, actually reinforce something that Robert Valdez said this morning, which is how much we appreciate the Center Fellows, who, as some of you know, but maybe not all of you know, that's why it's worth underlining, played a central role in conceiving of and planning and organizing the symposium. I also have a sort of egocentric thank you, because they sort of allowed me to contribute an idea or two during the planning process. In particular, they sort of tolerated and maybe even accepted my pressing for one element of the overall agenda, and specifically the one that, if you've seen the agenda, the theme and the questions, the wordy bit about reciprocal, non-reductive systemic relationships between contexts of health and varying levels of analysis, from the biopsychological through the sociocultural to the historical political. That's, I'm guilty of that. And the implicit, but probably obvious to those of you who know me, my press for the implicit, but now explicit press for greater engagement with inter and transdisciplinary perspectives and projects, which it so happens brings us to Sarah Gehlert. Because as you're about to hear, among her many noteworthy roles and accomplishments, and now I'm going to embarrass her, but that's okay, Professor Gehlert has directed and still directs several significant networks, and picking up on at least one of the discussion points before lunch, federally funded by NIH networks, by NCI, National Cancer Institute. So if you're wondering about, are these things becoming kosher? Yes, and Sarah represents projects that embody, express, develop research at the intersection of a range of disciplines and perspectives, and yes, guess what? Levels of analysis. Levels of analysis that someone might even talk about as hierarchical levels of analysis. Enough of that. Such research, I think, is rich both in its intellectual, dare one say scholarly and academic, I dare, rich in both its intellectual, scholarly, academic depth and elegance, a word you may not hear too often, and its social significance, or at least its socio-political potential. And the kind of research and work that I think demonstrates that those two dimensions, intellectual depth and socio-political significance, need not be mutually exclusive. Indeed, they can be creatively complementary as long as we don't fall into dichotomies as we tend to. And finally, because I think there's a non-trivial moral to the story, I'd like to tell you that I only became aware of Sarah's work about 4 months ago, when we happened to find ourselves at a conference on transdisciplinarity in, no, not Bethesda, not Boston, not Chicago, wait for it, in Taiwan. And there and then, as Sarah presented her and her colleagues' research, I thought, even shouted to myself, wow, this will be perfect for the Center's Fall Symposium, or at least the part of it that, part of the agenda that I've nagged and nudged and nudged about. And second, I said to myself, I didn't shout it, I sort of queried myself, how is it possible that as an ID-TD groupie, as a friend has called me, how is it possible that I didn't previously know all this brilliant cross-multiple boundaries work? In any case, perhaps saving the moral of that story for later, I'm both grateful that I now do know about Sarah's work, and having suitably lowered your expectations, I'm very pleased to welcome Professor Sarah Gehlert.
As an undergraduate at the University of New Mexico, and I've been reflecting on that. I haven't been back in 3 decades. But I realized I was studying biological anthropology, and there was a professor here named J.N. Spuler, who's been gone for many, many years. But he really piqued my interest in genes and social environment. And so as a 19-year-old, I developed that interest here. And ironically, I had to go back to the Midwest because my mother had breast cancer. So I'm here talking about breast cancer today, which is very meaningful to me. So in my 45 minutes, I want to use group differences in mortality from breast cancer as an example of how social inequalities change biology to create health inequalities. So going from social inequalities to biology to health inequalities. I've spent the last few years trying to understand with great academic and community partners the interplay between the social environment and biological factors. So that's what I'm going to talk about today. And I'm going to give you just a tiny bit of background. I'm going to kind of scope down to something very specific. But it's important to me to notice, to think through sort of the federal perspective on this, the federal process. So we really, when you look back, health disparities, health inequalities were first talked about in the 90s, but it was really the 1990 US Census that provided the first mortality data by ethnic group for all 50 states. It's the first time we had data on ethnicity for all 50 states, and that caused people to suddenly be alarmed. And the federal government had to respond. So there were many changes at NIH and other places. In 98, Clinton launched the Racial and Ethnic Health Disparities Initiative. Health disparities was named one of the two overarching goals of healthy people. 2010 continues to be, we haven't quite made much progress. And recently, and I'm going to come back to this, but data were available for health screening behaviors by ethnic groups, and I'll talk about how that figures in. But just to give you some background, and again, this is not the most recent, but as Tom said earlier, things haven't changed that much. So if you put all cancer types together, all groups, comparing across groups, you merge genders and you merge ages. You see that for incidence from cancer, African Americans had the highest incidence, followed by white Americans, followed by Hispanics, followed by Asian and Pacific Islanders, and American Indian Alaska Natives. So you move to mortality, and you see bigger differences. You see a bigger difference between African Americans, white Americans, but you see American Indian, Alaska Natives, Asian and Pacific Islanders, and Hispanics being more similar. We're going to talk about this. This is literally a very amalgamated view. So if we look at breast cancer, you notice, maybe this still won't show up. My laser pointer won't work very well. But if you look, we're starting at 1975 there, which is about when the National Cancer Institute started collecting statistics by race. And you can see that for mortality from breast cancer, black and white Americans or women were about the same till about the mid-90s. It was only in 1990 that we collected data from ethnic groups. So let's look at this in a different way. If we look at incidence, and I'm comparing black and white women because I was working at the University of Chicago, and this is the group with which we began working. A little bit different from what we saw earlier. White women are more likely to get, what's happening? I'm not even touching it. Okay, we have new equipment. And a better laser. Okay, so if you look at black and white women from 1973 on, you see that white women have always been more likely to get breast cancer. And the difference between the two groups has stayed about the same. But if you look at mortality, we noticed earlier in the mortality map that there wasn't really much of a difference between black and white women until about the mid-1990s. And then you started seeing improvement. For white women, the mortality rates got better, they went down, but you didn't see concomitant improvement for black women. So that nationally, black women, although less likely to get breast cancer, are 37% more likely to die from it. And is it biological? Well, let's look at how this plays out across cities in the United States. If you look at 3-year averages, 2005 to 2007, for mortality disparity, you see differences across cities. So we were looking at the US in the middle, which is about 37%, New York City, 27%, Chicago in the 60s. We have people in the audience from Chicago who can attest to this. Yeah, so obviously not biological. So the National Cancer Institute, in responding to this, because there was a mandate for each institute and center at NIH to come up with a response, developed transdisciplinary research initiatives, starting in 1999. So we've done 3. There's the Transdisciplinary Tobacco Use Research Centers, the Centers for Population Health and Health Disparities. I was the principal investigator of one of these. And then Transdisciplinary Research in Energetics and Cancer, which is basically obesity and cancer. And I'm the co-principal investigator on one of these, and I moved from Chicago to St. Louis. So I've been very involved in this transdisciplinary perspective on capturing disparities. So why transdisciplinary centers to address disparities? And these are NIH's words, by the way. The determinants of disparities are multi-level, complex, and their interrelationships are poorly understood. You need synergy across the biological, clinical, social, and behavioral sciences to address this complexity, to capture it. And you need to train a new generation of transdisciplinary researchers, which is something we talked about a little bit earlier. So in 2003, the National Cancer Institute started the first phase, the first group of Centers for Population Health and Health Disparities. And they were directed at cancer disparities. There were 8 centers in the United States, each with at least 3 interdependent research projects addressing biological, behavioral, and social aspects of health. And 1 project at each center had to be community-based participatory research. So it was a new way of looking at things. And for the first time, not only did they have academics working with community members and disparate disciplines working together, but they also tried to get the institutes and centers at NIH to work together. That didn't work terribly well, I'm afraid. So it's in the second round now. And this is a report that we published in 2007. It was 8 centers who were looking at different types of cancer. They were looking at different genders. They were looking at different groups. So all of them kind of took this approach. They had what they called an incubator, a kind of neighborhood community context in an incubator of community engagement. And they went from biomarker and animal model studies. So that was to look at genetics, biological basis of disparities, biomarkers of risk assessment and screening, to preclinical and clinical studies, looking at etiology, screening, and treatment, to impact on patient and community, dissemination of research, quality of life, access to care, and policy. This was a pretty broad, pretty ambitious initiative. So what do we know about the causes of health disparities? Before I talk about specifics of my center, I want to just sort of bring us up to speed here. So what factors potentially influence cancer risk? Well, macro level factors, regulations, policies, societal views, genetic factors, inherited mutations, sporadic mutations, telomere now, individual level factors, diet, physical activity, health behaviors. Meso level, we now have some work to suggest that the nature of early family relationships is connected to developing breast cancer in adulthood, and demographic factors. So the point is, I think that the reason we hadn't made more progress is that we tried to sort of do these individually, and we didn't take the bigger picture of how they influence and how they interact with one another. So the group of 8 centers put together this scheme, predicting cancer disparities, or the determinants of cancer disparities, and it was published in the American Journal of Public Health. And we would redo it now. You know, you publish something, you're stuck with it, and people say, and we realize it's not great. But we sat down as a group of centers, and we had 38 different disciplines, and we tried to come up with what was the multi-level scheme. So we went from social conditions and policies, culture, norms, racism, sexism, discrimination, public policies and poverty, to institutions, healthcare system, families, churches, legal system, media, political system, neighborhoods. We looked at collective efficacy of neighborhoods, social capital. You could argue about how similar they are. Access to resources, neighborhood stability, social relationships, social networks, social support, social engagement, individual level, and biologic genetic pathways. So across these 38 disciplines, and across each center, but mostly across all the centers, we covered the bases and tried to work on these models. So the scheme that we used, I mean, there really aren't a lot of schemes, but we sort of adapted Mike McGinnis and his colleagues' scheme of the multi-level determinants of, we use this for health disparities. Actually, it was a meta-analysis of 500 studies on early deaths in the U.S. So McGinnis and his colleagues thought that roughly, just as a way of starting out, about 40% were due to behavior patterns, maybe 30% to genetic predisposition, 15% to social circumstances, 10% to shortfalls in medical care, and 5% to environmental exposures. And they were talking about environmental toxins, not social environmental exposures there. So it was really the social circumstances, social environment that we thought had been given short shrift. So just looking at what we mean by these, you know, behavior patterns, we were talking about with genetics, heritability and genetic propensity. We were talking about for social circumstances, discrimination, availability, distribution of services, shortfalls, bias. We talked about the Institute of Medicine report and exposures. And when you tried to map those across our scheme, it looked a little bit like this. So we had a frame that we were trying to investigate. We were trying to sort of take a view of cancer disparities from up in the air. And we think that the reason we've made so little progress today is that we focused on behavior. We focused on the individual level and genetics to the exclusion of other determinants, especially social circumstances and shortfalls in medical care. If you looked at, I didn't do a slide, but if you looked at the NCI dollar and what's funded, I can promise you that most would be done to change behavior patterns. And we've also neglected, importantly, the interactions between these determinants. The proof is in the interactions. So how living in poverty affects health behavior, how genetic propensity might affect response to exercise. There's a new field of endogenomics I've learned about, which says that some people, by virtue of ancestry, just don't get the pleasure from exercise that others do. So getting mad at somebody because they don't enjoy going to the gym really isn't fair. You need to find something that will work for that person to address obesity. I fortunately really get the endorphins. I love to go to the gym. So. But also we know in terms of nicotine addiction, in the last year there have been studies that some people from some ancestries are much more prone to addiction than others. So, is it health behavior? I just bring this up because whenever we also talk to medical students each year, and I always ask them, which group, racial ethnic group, has the lowest rates of screening mammography? And their hands always go up and they say, African Americans, we're sure. Really not the case. Not that screening mammography solves the problem, but it's simply, I'm saying this because you don't want to get locked into a mindset So actually, African Americans, as of January 27th of this year, had the highest rates of screening mammography. And Mexican Americans and Filipinos have low rates. So why are we pouring resources into increasing screening mammography for African Americans? We need to just be aware. Let's see. So is it just genetics? No, because a low percentage of cancers are thought to be caused by hereditary mutations, a fairly low number. So 5 to 10% of breast cancer, 10 to 15% of lung and prostate, 20% colorectal. A higher percentage are due to sporadic or acquired mutations that happen across the life cycle, and these happen due to environmental and social exposures. So, social circumstances are the least studied determinants, but they're potentially the most significant. They're perhaps also the hardest to study. So, this is sort of Tom threw one out to say this is kind of the way we've been taught to look at things. So if you just look, this is life expectancy at birth. If you just look at white, black, male, female, you get the pattern that probably the medical students are used to. You get the highest life expectancy among white females, you get the lowest among black males. But if you begin to include social factors, you see a different picture. And I don't want you to think I'm falling into what I'm now calling the Leveist fallacy, because I'm not. I'm not using this to compare across races or ethnic groups. I think that's good. We can start calling it the Leveist fallacy. And this is something that Mira and her colleagues published in 2008 in Health Affairs. And and look who walks in. I was talking about the Leveist fallacy. So this is looking at change in life expectancy at age 25 by race, gender, and education. So we throw in one social variable. So it's a change from 1990 to 2000. And Mira and her colleagues took the members of this group who had the highest levels of education and the lowest. And it suggests that for people with higher education, you see the pattern that we saw in the last slide. But if you look at the low education group, it's a different pattern. So the least gains have been exhibited by white females. And again, I'm not comparing across races. I'm simply saying that adding a social variable makes a difference. So, and Mira suggests that high rates of obesity and smoking, the smoking rates of lower SES white females are very high. Not sure. Also, this is the slide that you were talking about earlier from Health Affairs. This came out a couple weeks ago. And just again, not comparing across races, but looking at education. So this is less than 12 years of education, 13 to 15 and 16 or more, 1990, 2000, 2008. The point is, life expectancy is much lower. And this is only white females. But life expectancy is lower for women, white females who have lower levels of education. And education, it's changed through time. So that the situation seems to be worse, or was worse in 2008 for women with less than 12, white women with less than 12 years of education. Okay, this is one that I'm still having trouble with. I just took this out of the New York Times got the article about 4 days ago. My point again isn't comparing across races, but that lower levels of education for every group are associated with lower life expectancy. So again, a social variable changes the picture. So I'm going to give you an example of our Center for Population Health and Health Disparities at the University of Chicago. I moved from the University of Chicago about 2 years ago. This work is still continuing there, and we're working in neighborhoods in St. Louis and in Paris, actually. We've been working with French-speaking African immigrants to France with breast cancer in Paris. So this is our center. We were a group of scientists who'd never worked together before. We were at the University of Ibadan in Nigeria. We were at the Cancer Risk Clinic at the University of Chicago. And neutral space for us was the Institute for Microbiology at the University of Chicago. It was the only place that wasn't part of the med school or social sciences. So it was neutral space where one discipline wouldn't be privileged over others. It was administered not by any one of our departments. Okay, so I was from the School of Social Service Administration. I'm an anthropologist and a social worker. Martha McClintock is a biological psychologist. Suzanne Konzen is a breast oncologist who's also a molecular biologist. Fumio Lopade is a geneticist who's also runs the high-risk breast cancer clinic at the University of Chicago. And Tomasz Kraus is the chair of surgical pathology. There were also about 30 other people, but these were the project leaders. So we'd not worked together, 2 schools and 1 department at the University of Chicago. So the question that brought us all together was, what factors in women's social environments contribute to the African American and white disparity in breast cancer mortality in the U.S. that I showed you earlier? And it's interesting, this was the common ground. So the clinicians had always wondered, they knew that medicine wasn't going to do it. They knew that no matter how good the treatment became or drug discovery became, there were still going to be differences. And they were pretty sure it was social environment. So when we talked about this, they said, I'm in. And the pathologist, interestingly enough, said he was in because he'd spent 30 years looking at women with breast cancer through a microscope, looking at their cells and always wondering what was going on with the rest of their lives. So I think a question that is big enough for everybody to be part of brings disciplines together. So how much of breast cancer is hereditary? As I told you, not very much. About 5 to 10% is due to known hereditary mutations, BRCA1 and BRCA2. These family clusters are families where we know there's a lot of breast cancer, we just don't know why yet. And most is 70 to 80% is due to just sporadic mutations in the breast cancer gene. So the breast cancer gene protects you against breast cancer, and if you knock it out, you don't have that protection. So as we're all sitting, we're having little mutations. But in a healthy body, one that's actually got to some sleep, you repair. But if you're living in a social environment that's taxing you constantly, psychologically and physically, you're prone to these mutations. So our model was, when you got all these disparate people together, that race doesn't equal biology, it determines the social circumstances in which one lives in the United States. Which leads to certain psychological states, which leads to neuroendocrine and gene expression changes, which leads to the survival of these mutations, of these mutated cells that grow into what we call big whopping tumors. So we were going from social process to genetics, and it took all these disciplines to be able to test this model. So I was obviously way up here, Martha was here, Crouch was down here. So we put together this scheme, mutually informative, multi-level and multimodal approach. We were in Ibadan, Nigeria, and we were on the south side of Chicago. I'm just going to talk about Chicago. We had 2 projects that used animal models. And the reason they used animal models was you can't vary the social conditions of women, nor would you want to. So the animals live about 800, 900 days. You can take a whole life cycle perspective, and you also can vary social conditions. And then Fumi and I worked with 300 women who were kind enough to volunteer for the study at the point at which they were diagnosed with breast cancer, when they were being treated at either the University of Chicago Hospital, Stroger Hospital, the public hospital, or Mount Sinai. So together, we went from looking at community, neighborhood variables like crime, collective efficacy, social ecology of neighborhoods, to housing, environmental exposures, to social circumstances, this one's for religious, social isolation and social support caused by these factors, to psychological states and behavior patterns, to hormones and genes. And it's the hardest thing I've ever done. With community members helping us all along the way. And you think, how different could you be doing animal model work and community-based participatory research? Bob Hyatt at University of California, San Francisco makes this happen, and they've actually produced a video with community members called Of Mice and Women, explaining how using animal models and this kind of science is important and can change the health of women. So I'll give you a little bit of this animal work again. I'm a social worker anthropologist. I've never had animals before. But Martha uses them. She never harms them. She just varies the social conditions. And this is a natural state of these female mice. They pile on top of one another. They groom one another. They rear one another's young. And they're a good model of human disease because they're social like humans. So this one here is in the preferred position. She gets to stay there for about an hour and a half, and then she'll rotate and somebody else will go there. So through the day, they vary. So what Martha did was, because based on work by John Cacioppo, who some of you may know, we thought that social isolation was really an important factor. So Martha would take one rat at different points in the life cycle away from its sisters and isolate it. It would be in the same room, same food. It would be in a cage next to its sisters. Everything would be the same. It would just be isolated. And she found that the greatest influence was if it was isolated during puberty. So what she found was that the isolated animals over here, compared to the ones that were left with their sisters, literally sisters, it would change their behavior. So we're looking at the cage from the top, and this is a novel stimulus. It's an overturned food dish. And usually animals, when a food dish is overturned, will try to get to it, figure out what it is. The blue is just tracking the footprints of one animal for 24 hours. So what we see is that the animals left to their own devices do what's really adaptive. They explore their environments. They get to know their environments. They walk around. This is the rat. But the animals that are taken away from their sisters, from their peers, begin to just stand in a corner and look out for threats the whole time. It alters their behavior, and they end up living about what would be the equivalent in human. They live about 200 less days than the group house. So this has a real effect on behavior. And it's really interesting. I was working with, NPR had me talk to high school students from the South Side, and a kid from Paul Robeson High School. I was presenting this to them, and he came up with the best question we ever had, which was, if you put the animal back in its group, what happens? Does it reverse itself? Does its health improve? So I said, good question. I went to the cell phone, called Martha, and said, we gotta put them back in the social groups. Those kids are the best idea ever. And it really turned our science around. So I honestly think that CVPR enhances science. So what we found was that the isolated ants, a little hard to look at, but the group house animals look like this. And the sister, which is genetically similar as possible, has, through isolation, developed these tumors, these big tumors, and quit grooming herself. And then when you look at the tumor burden of the isolated animals versus the ones we left alone, basically, you find 83 times more tumors in the animals that are stressed socially. So, and first, we couldn't figure out if they'd be malignant. The pathologist said, oh, they won't be malignant. So we asked NCI. We said, can you give us a scheme for figuring out, tumors in rats? And they said, I still have the email, Dr. Gehlert, you're on your own, but when you find it out, let us know. So, we were working with Annette at that point. So Suzanne Kanzen, this other young scientist, she said, well, what the heck's going on? How's the social environment getting under the skin to change the biology and produce these tumors? And we started looking at the stress hormone levels. So we just moved the animals' cages, the ones that were with their sisters and the ones that were alone. We just moved them. It was really a mild stressor. And we measured the stress hormone levels in the blood before we moved them, after, 30 minutes after we moved, or 90 and 150. So we looked at how fast that stress hormone went up and how long it took to recover. And we saw big differences between the animals we left alone and the ones that were isolated. And that's important because being exposed to those stress hormones is really, really bad. And one of Martha's students was able to predict the development of tumors, whether the animals developed tumors by how far up it went, and whether they died or not by how fast it went down. So we knew we were onto something. So we then took this information, we went back and forth with the animal models to the wonderful women who were working with us who had breast cancer. So many women agreed to be in the study, it overwhelmed me. They really were incredibly anxious to figure out any way that we could change the high rate at which their mothers, grandmothers, sisters, daughters were dying from breast cancer. So we focused on these 15 neighborhood areas in Chicago because we wanted to go in depth. We knew that place had a lot to do with it. So we wanted to be sort of circumscript in the place. We wanted to gather lots of variables, the whole multi-level model. And that's where the University of Chicago Hospital is at Lake Michigan. So small geographic space began to cause transdisciplinary problems because Fumio Lopate is an oncologist, and she said it's got to be case control studies, 20,000 women. I said, we can't learn about neighborhoods in place with 20,000 women. We've got to go down to 300. So I finally won that one. But I was so mad when we were arguing one day. I was half a mile back to my other office in January in Chicago, and I realized I'd left my coat at the office. It was probably like 20 degrees, and I didn't even feel it. I was so upset. So that's the perils of transdisciplinary, or the beauty of transdisciplinary. So this is a way. So we began working with the 300 women, and when they went in to have their surgery, Fumi's lab went in and took excess tumor tissue so that she could look at the biology. And my group began following women in their homes, and we used master's level social work students who were African American and had grown up on the south side of Chicago. We were going to follow women in their homes. We did interviews in their home, and this is, we have many, many, many things that we follow. I'm just giving you examples. We looked at psychosocial functioning, social network, health behaviors, perceived discrimination, and we collected salivary cortisol, daily total reactive stress, and then allostatic load measures. We were lucky enough to be in Chicago. I don't say that sentence often, but because they collect data on crime. And we were able to get really good data on crime so that we could construct quarter-mile buffer zones around each woman's primary dwelling, if they were homeless, where they spent the most time. And then we could compare crimes across women. We also had measures of how safe the housing was in Chicago. So we were able to sort of model that, that vigilance, acquired vigilance that we saw in the animals. But we also had collective efficacy. We used some of Rob Sampson's data also. But it was still too, even that quarter-mile buffer zone was just, and these data were not close enough to women's homes. We found out that it was really what happened right around them that mattered. So we added another level, neighborhood. I had to learn about built environment quickly. So we started mapping the 4-block radius around each woman's primary dwelling. And we looked at some of the things that were talked about earlier by Dr. Levy's, liquor stores. We looked at health facilities. We looked at traffic. We looked at many, many things. We found a lot of vacant lots, which I'll get back to, and we had to develop a metric for the vacant lot. Was it a place where people could socially interact, play softball, or was it full of syringes? Did it really change the whole character of the neighborhood? We actually, at one point, tripped over a bag of medical waste that somebody had just thrown off an ambulance in Englewood. People just consider some neighborhoods sort of sewers. And we looked at vacant buildings, too. So what we found was that there was a high correlation between what was happening right around where a woman lived, her response to it, and what kind of tumor she had. So we did this satellite mapping of each woman. We sent in students to map the neighborhood and come up with our metric. But I had on my desk one day this, which I showed you earlier, and these things. And it occurred to me, that in the same way we manipulated the social environment to produce the tumors in the animals, that the social environment was manipulating what was happening to women to produce tumors in these neighborhoods. Now, not all the neighborhoods, we were in Bronzeville where the Obamas have their house, you know, and Minister Farrakhan lives, to Inglewood where things are really, life is not easy. It's where Jennifer Hudson's family was killed. So one thing we looked at was this diurnal salivary cord, which is measures reactive stress. What's happening to the woman, what her body is telling us is happening to her throughout the day. So we measured it when she woke up in the morning, half an hour later at lunch and at bedtime. So this is 2 days. We kind of looked at this nighttime rise. And we found that women who were living in who reported the most stressors had this high shoot up of cortisol to try to deal with the next day in some, but I'll show you. We call that the oh shit response. You get up and you're ready to tackle the day, you look out the window and absolutely nothing in your neighborhood has changed. So we started, as an anthropologist, I used to do cluster analysis, which is way out of fashion. But we cluster analyzed these salivary cord patterns, and this was just overwhelming to me. So we found two groups of women. Some that were acting like the stressed animals, their stress levels were going up, but then 67% of the women in the study weren't reacting at all. And when we looked at who these women were, we found that they were the ones who in interviews had, when we asked them to list the 4 most negative things that were happening, we had them do positive too. They're the ones who put breast cancer 3rd or 4th on the list. Housing, food, safety was at the top of the list. Breast cancer just wasn't even an issue. And they were the women also, we did follow-up at 6 months and 12 months who were hardest to find. So we found, and I wasn't going to promise right that we would do too much biology, but we found this flat cortisol pattern was an independent covariate of having triple negative breast cancer. And triple negative, we weren't trying to predict whether women got breast cancer or whether they died from it. We were trying to predict if they got this triple negative breast cancer that's associated with higher mortality. Do you know what triple negative breast cancer is? Probably women do and not men. Triple negative breast cancer is something that you see in West Africa and North Africa a lot. It's a premenopausal kind of breast cancer. We see kids who are 16, 18 of West African ancestry with breast cancer. And not only does it happen early, not like the pattern that we see among women of European ancestry, but it's particularly lethal and aggressive. So it's important because that finding was triple negative breast cancer is most common in African American women, followed by Hispanic women, although NCI needs to do a lot more work here. And African American women are 3 times more likely to be diagnosed with it than white women. No figures for Hispanic. And mortality from triple negative breast cancer is twice as high as non-triple negative breast cancer. This has many, many policy implications because if it happens at 16 and 18 and they recommend screening starting at 50, good idea? I don't think so. So just to give you, I wasn't going to also give you a lot of results here. We're able to predict the triple negative from the neighborhood variables, but here's just a logistic regression from the social level variables that I gathered, robberies, homicides, to the psychological depression. We were able to predict which of those 2 groups women were in. It's a pseudo R squared, but about half the time. So, you know,